This week in lab, you're going to be planning an experiment. The goal is to determine viable cell counts needed to calculate the volume of a yeast culture used for brewing beer or wine. But the very first thing that we need to do is determine how many cells are in a vial, and we need to do dilutions to do that. We'll focus on these skills, working with large cell numbers, determining volumes needed for simple and serial dilutions, and the calculations for that. This is what a yeast culture looks like. It's very dense because there are billions of yeast cells in this culture. So to do the experiment that we want to do, we're going to have to dilute this culture. We're going to dilute those yeast cells so that we can have manageable numbers of cells to work with. What we're going to do right now is simply plan the experiment. And the first thing that we want to calculate is the dilution factor. Notice that we're starting, we're going to work right now in column 2, and that's Eppendorf number 2. We have predetermined that we want 750 microliters of yeast, the culture that was shown in the previous slide, and 250 microliters of water. We add to that, and our total volume is 1,000. Now, if you look up here, the definition of dilution factor is the volume of the item being diluted over the total volume. So let's ask a couple of questions. What is being diluted? Well, it's the yeast cells that are being diluted. And we've got 750 microliters of the yeast cells. What is the dilution factor then? Well, we've got the 750 microliters of yeast. What's the total volume? We're going to add the 750 microliters of yeast plus the 250 microliters of water, and that gives us our total that I just mentioned, which is 1,000 microliters. So we've got the yeast, 750 microliters over our total, yeast plus water. We can simplify that to 3/4. And what we're going to do with that is simply plug that number into our table. And in just a second, you'll see what we're going to do with that. Now we're carrying forward the information from the previous table, and here's our dilution factor for tube number 2. Next, we want to calculate the number of cells per mil in each tube. So we want to do tube 2 now, and we're going to assume that our starting culture contained about 5 times 10 to the 8 cells per mil, and that's shown here. Later on, you're going to actually work in the opposite direction. You're going to start with a low number of cells that you determine from a plate and extrapolate back. We're simply showing you how to do the calculations in the forward direction right now. But they'll be very similar. It's whether you're going one direction or the other. So to do this, the sample was diluted between tubes 1 and 2, so there are fewer cells per mil in tube 2. Well, how do we determine how many cells? Well, we're going to multiply the 5 times 10 to the 8 by 3/4 because we have only 3/4 or 0.75 the number of cells that we started with. So if we do that calculation, 5 times 10 to the 8 times 0.75 or 3/4, we end up with 3.75 times 10 to the 8. Notice that we're not really changing that exponent very much. We're really just changing that number a little bit. We've still got billions of cells, almost billions of cells. And now we're going to plug that number in here. And we can keep working this table until we do everything just doing it mathematically. Well, we just calculated that there were 3.75 times 10 to the 8 cells per mil in tube 2, and we've entered that into our table here. How accurate is this number? And how many of those cells were actually alive? Some of them might have been dead. Ultimately, to find these types of answers, you're going to spot known volumes of that tube onto growth medium and see what grows. And now at this point, we can actually calculate how many cells we might see, how many cells will grow into colonies if all of them grow, if we spot 3 microliters on a plate. It's called a spot plate, but it's medium that will support the growth of the yeast cells. So now we're going to spot 3 microliters. We actually have a culture in which we've got it listed as per mil, so microliters is only 1/1000th of a mil. So we're going to factor that in. So now we've got 3.75 times 10 to the 8 cells per mil times 3 microliters, but that's actually equivalent to saying 0.003 mils. So we're going to get rid of the microliters and make sure that we're using units, mils, that are consistent between those two numbers. And what we end up with is roughly 1 times 10 to the 6 cells, because we've canceled out the mils. We're going to cancel out. And we're only showing significant numbers here. And now we can plug that into our table. Now that's a million cells. You're going to see a big blob of growth where you have spotted that 3 microliter aliquot after 24 to 48 hours, because there are so many cells you will not be able to count them. But that's why we do dilutions, because ultimately you'll have a dilution that's high enough that you will have countable numbers of cells. Now we'd like you to try to finish the remaining calculations for the simple and serial dilutions in the handout that's posted on BB Learn. If you can do it before you come to lab, then that would be a good thing, but at least before you leave the lab this week.